Okay, so the title of this talk is Noxious, Invasive, and Near Endangered Evolutionary Changes in Weed, Integrated Pest Management of Beetles and Soil Contaminants and Monarchs. So this talk's going to cover a wide array of different spectrums from population genetics to integrated pest management, as well as mechanisms of, or my new direction where I'm looking at elements of phytoremediation. So as you all know, there are probably going to be more species being threatened, endangered, or extinct over the next few hundred years. And this growing number is on a range from least concern to threatened. But first, it depends on whether or not we have the data to even make such a claim that a species is at risk. And once you have the data, you could evaluate it. And if the data is adequate, you could put species along this range of extinct, critically endangered, or vulnerable, as well as a bunch of other different categories that might be pertinent to rescuing a population. So a bottleneck has to do with population size and its decline over time. And after a population experiences a bottleneck, it is reduced here to this valley where it can either be recovered or it could go into extinction. Now, this is particularly dire in some circumstances. If a population does not recover, it'll go to an extinction. And in some cases, when a population recovers, the genetic diversity that was once in a fully functional population is lost. So here you can see, here's the original population, and you can see that in the surviving population, there is an allele, which is represented as the green circle, is missing. And so this is what you could have due to a bottleneck effect. And I know sometimes you all see this from some of your textbooks and you're like, yeah, you know, bottleneck effects, and you think that they don't really appear in nature in real life again. But that was not the case for the Florida panther, which was reduced to about 6 individuals, including one mother. And this could be captured with mitochondrial DNA, which is inherited maternally. So previously to that bottleneck, there was 2 other bottlenecks, so 3 bottlenecks in total. So bottlenecks are drastic, but in some cases, populations could bounce back, and this population was able to be recovered through the introduction of a Texas panther. But when species are drastically declined, the survival probability also declines. So here's a paper also from the 90s that was looking at the survival probability of the North Atlantic right whale. For those of you who are not familiar with the history of New England, that was the primary source of New England economy, was whaling. So rightfully so, some of these marine mammals were depleted and also had experienced bottlenecks. As it relates to this region here in Kansas, in this growing direction, my collaborators and I were interested in the growing decline of monarch butterflies. And so here is monarch count data from the Xeres Society annual monarch Thanksgiving count. And you can see that over the last 20 or 30 years, this population is being depleted. And as I showed you, at this rate, if this continues, maybe we could find a way to mitigate and rescue this population. But after there's a bottleneck, some of the genetic diversity in alleles may be lost. Now, on the opposite side of the coin, whether it's an endangered, threatened, or near endangered species, you have the antithesis of endangered species, which is the invasive, right? So these ones do not make the National Geographic cover. So invasive Colorado potato beetle has a range expansion that is intercontinental. It originated in Mexico, and then it spread intercontinentally. So the outline of this talk, what I'll mainly be talking about is I'll be weighing and I'll teeter back and forth between noxious and invasive species versus threatened and near endangered species. And so the 3 research questions that I'll be mainly addressing is genetic diversity. Has noxious weedy chicory changed since its introduction? Then I'll be talking about integrated pest management of the invasive Colorado potato beetle and looking at whether or not microbial inoculants can be a form of viral protection to deter pests. And then I'll be talking about my new research direction, which is environmental toxins and how it may affect monarch behavior. So beginning with chicory, chicory was first introduced into the United States by about the 19th century. According to Thomas Jefferson's journal, he received the seeds from George Washington and he planted them on the Monticello as a way to feed sheep. And then these populations were all throughout the East Coast. In 1785, it's documented that Governor Bowdoin of Massachusetts also planted it to feed the sheep. And within 20 years, the population of chicory, Cicorium intybus, was naturalized throughout the 13 colonies, so on and so forth. By 1818, it was abundant in Pennsylvania and in around Philadelphia. So originally, when we first came across this research direction, this was work that I developed with my collaborators at UMass Boston. I had collected some of the original specimen that we stored and it turned into a life of its own. We were interested in multiple versus single introductions of chicory, just playing around with the data. So here is a paper that was released in 2017 that we're able to know that there was multiple introductions of chicory into the United States. To orient you, here is Sicily, here is Italy, that's Northern Africa, and here is Ukraine. So this is a white and black map. And you're seeing that there is two haplotypes based on chloroplast sequencing. And you can see that the populations are on sort of this northwest, southeast divide. Populations in Germany, Czech Republic, Poland, and Russia have this blue haplotype, while Sweden, Hungary, Yugoslavia, and Iran have a different haplotype. And you can see that in the chicory's introduced range that all these haplotypes are represented, but you see this third haplotype here from the sample we collected at Monticello, and it's a bit different. It could be different due to introgression or just outcrossing with some of the native flora that was already here for whatever reason. But here in this experiment, what we're actually interested in is the change or the microevolution in chicory over time. So that's over the last 200 years since it arrived into the United States. So to go about this, we compared DNA fragments of herbarium specimen with those of contemporary populations. And the methods here is getting the plant material and extracting the DNA. So when I first began on this project, it was hard to extract DNA from herbarium specimens. So when you have old museum samples, one way to do it is with modify the protocol and use liquid nitrogen. And so for the plant material, there were herbarium specimen, 84 individuals, including the contemporary populations, 18 contemporary population, 8 specimen per population, for a total of 228 individuals. And then for markers, there were 12 nuclear markers. Nuclear markers were microsatellite markers, which are 3 simple sequence repeats in the DNA, so CAG, CAG, CAG. And then we're able to analyze the fragment lengths to tell us about the population structure of these species. And also there were chloroplast markers. In this study, we identified 3 haplotypes through chloroplast sequencing, and this allowed us to do DNA fingerprinting. So what we used here was a forward and a reverse primers, which is amplifying these repeats. And then there was a third primer, which is this M13 fluorescent primer, which allowed us to look at fragment lengths at, here you're seeing locus 1, 2, and 3, but we did it for 12 different loci. And so we analyzed it on a software called Peak Scanner, and the way Peak Scanner works, and this is actually from some of the original data. Talking with my collaborator last night, we saw that there was a date on it that was so old that we had to remove it from when we was playing with it. And then, anyways, the 2 peaks here, big peak, little peak, are the heterozygotes observed, and big peaks here are just the homozygotes, and this allows us to analyze the nuclear DNA, which are the microsatellites. As for the results, what we saw was that In the herbarium specimen, the 3 haplotypes were observed in both the 19th and the 20th century. So these are the specimens that predated in the 1930s. You can see that all 3 haplotypes are here. And you can see that by about 1940, the haplotype 3 is becoming increasingly represented, but all 3 haplotypes are here. And the main take home here is that haplotype 3 was rare in the historical populations. As you can see, it's only here in Connecticut, but in the current populations, we're able to see more of haplotype 3 here in Vermont, here in Haverhill, Dover, and on the Cape and the islands. And so you're seeing that there's these this microevolution happening in chicory since its introduction. We're looking at structure. Structure is a software that assorts groups based on genetic similarities. Each of the columns represent an individual, and it's assigning them colors based on genetic similarities. You can see that the H down here is the historical population. Those are the herbariums. And you can see that the herbariums are mainly different from the contemporary populations, except for 2 of the 18 contemporary populations showed similar genetic similarities with that of the herbarium. When looking at this through a Pico A, in your northwest quadrant, you can see your herbarium specimen, and each of the points here represent one of the specimens. The ellipsis represents 95 confidence intervals, and you can see that the herbarium are all assorted together, whereas the contemporary populations, which are color coded with the ellipses, they're not really overlapping with the herbarium, and this is further strengthens the point that the evolution of the population has changed, and we are able to observe the microevolution. So in conclusion, microevolution was observed in chicory. Herbarium specimens form unique genetic clusters, and HAP3 is more frequent in contemporary populations. For the next part of this talk, I'll be talking about integrated pest management. Can the invasive Colorado potato beetle be mitigated by soil inoculants, mycorrhizal fungi? So here's the specimens that I was working with in the Department of Entomology at Penn State. These beetles are all oligophagous. They feed on the family Solanaceae, ranging from tomatoes to potatoes. Most of the damage is caused after adult emergence. And as I mentioned, they now have an intercontinental range that expands from the Americas to Eurasia and as far east as far Russia. And one important thing to note is that these specimen Colorado potato beetles are hard to eradicate. And here you can see some of the genetic diversity. Interestingly, here is Kansas. Kansas is the pink represented here for these different haplotypes. You can see that there's haplotypes that are just only pink, meaning that in Kansas, we have Colorado potato beetle haplotypes that are just unique to the state of Kansas. So back to the Colorado potato beetle invasion. Colorado potato beetle was here in between Nebraska, Kansas, and Missouri by 1859 and 1862, just on the brink of the Civil War. By 1876, it had made its way up to New England sometime near the end of Reconstruction. And by 1960s, you can see that these Colorado potato beetles had made their way into Poland and into Ukraine and Russia, and already had made its way intercontinentally. In terms of dealing with Colorado potato beetle, they have introduced pesticide resistance, such as organophosphates, as early as the 1930s, and they were widespread and used, and especially to eliminate Colorado potato beetles by 1964. But because these species are highly diverse and highly adaptable, they actually developed to become increasingly resistant. And this is due to 2 point mutations on a gene that codes for acetylcholine esterase. And it's basically a point mutation that turns serine to glycine. And these beetles are increasingly resistant due to that. So here's from the paper, here's the y-axis. And the reference is from the similar paper I showed you, the haplotypes. So I should put that on there if you're interested. So here's the frequency of this mutation where you're going from serine to glycine, and you can see that the populations that are the frequency of the black and gray are the resistant individuals. So here are the homozygous dominants, and here's the heterozygous individuals, and they are also resistant to the pesticide. The ones that are not resistant are the white squares or the non-color filled squares. So there's more resistant Colorado potato beetles out there, and they're highly adaptable. So the aim of this project was, because we know that the Colorado potato beetles become increasingly resistant to the pesticide, we want to know if mycorrhizal induced resistance can play a role in providing bioprotection. Mycorrhizal induced resistance has been shown in other systems to protect plants via direct competition with intracellular root pathogens such as Phytophthora parasitica. Also, mycorrhizae can protect plants by altering the root exudates that's being sent out in the soil as chemical cues. Also, they could enhance tolerance due to increased lignification of the roots, but in, say, the above ground, mycorrhizal fungi can lead to what you call induced systemic resistance. That's the ability to protect the plants at very distal tissues by inducing genes in the salicylic acid defense pathway versus those in the jasmonic acid defense pathway. So because of this, it was interesting to know whether or not mycorrhizae can protect tomato. In a previous study, I found that mycorrhizae was able to protect against a gram-negative pathogen that uses type 3 secretion systems to inject infectors and plague soybeans. So this was sort of a follow-up project to that. So do mycorrhizae increase resistance to Colorado potato beetles? So here is the first analysis. We had 6-week-old tomato, and you had plants that were present or absent for mycorrhizal fungi. And after 96 hours, we were able to score the damage through image analysis using image J. And this was done for neonates. So these were beetles that had just hatched from the egg the night before, and they had nothing to eat. So it was a race against time for them to not dry out in the sun versus overcoming these trichomes on the plant and trying to get access to the resource. Versus some of the older beetles, which were third instars, we weighed the biomass, which was just the initial and after, and using the proportionate increase in mass. And here's the nylon meshes that was used to enclose the herbivores to the plants. What we found here was that plant tissue consumption was actually increased by AM fungi. So this suggests that mycorrhizal fungi did not provide bioprotection. The mechanism at play is likely to be plant anatomical defenses, such as trichomes. So trichomes are sort of like these protrusions that just are able to protect the plant to buy some time so they're not completely defoliated. It turns out that in plants that were inoculated with mycorrhizal fungi, there were less trichomes per unit area. So that's likely to explain why the plants were able to eat better with AM+, because AM+ was also reducing the trichomes. And this was observed across a broad array of different mycorrhizal treatments. So here's your control, no AM fungi, and then there's more trichomes. And in these inoculated plants, there were less trichomes. And this pattern is also seen when looking at the plant tissue consumption. Focusing on the graph on the left, you can see that it's obvious that plants that ate more, beetles that ate more on plants survived at a greater rate. And there was no correlation between the third instars and survival. So this is likely be the case because by the time the beetles make it to third instars, they're really large, and trichomes are like very small compared to a large beetle versus a newly hatched neonate. So AM fungi did not provision bioprotection. AM fungi promoted survivorship in early stage beetles, but this suggests that AM fungi can promote trophic structure. So that means that if the primary consumers are able to do better, so will the tertiary consumers, and this will allow there to be better trophic structure. As for the next portion of the talk is looking at the threatened and near endangered monarchs. So as mentioned, these species are on the decline, and there are many reasons why they may be declining, whether it's various forms of abiotic stressors or loss of habitat. But one of the forms of abiotic stressors is herbicide use. So just spraying plants with herbicides is a way to harm the monarchs, as some research has shown. So this suggests that monarchs are just highly sensitive to negative anthropogenic induced effects. And so this can also be seen with something such as zinc. So in this study, they had media that was either depleted or non-depleted from macronutrients, and they increased zinc toxicity. You probably didn't know zinc was toxic, but the monarchs are highly sensitive. You can see that when the macronutrients are depleted, you can see that the superoxide dismutase, which is enzyme that converts the superanion oxide into hydrogen peroxide, and hopefully catalase could then convert it to water before it becomes a hydroxyl radical, which could cause DNA damage. So you're seeing that they're looking at these antioxidant genes, and you see that these antioxidant genes in the monarch are being induced when there's zinc toxicity. So monarchs are highly sensitive to various forms of anthropogenic activity. This probably suggests that monarchs are also sensitive to other forms of anthropogenic activity, such as petroleum manufacturing, the burning of fossil fuel, which can lead to gas particulates and long-range transport. It can be deposited as these PAHs, which are polyaromatic hydrocarbons, and they could get into the environment through many ways, through more ways than one. And so here are the different forms of PAHs. They're characterized as having these multi-aromatic rings, and they occur naturally in coal, crude oil, and gasoline. Even the burning of just, if you have a fire pit at night, you're probably contributing to the problem. But Kansas is rich for this because we have a lot of oil and gas fields, so we have a lot of sources of PAHs in our environment. So for the methods, we focused on this PAH in the lab, phenanthrene, and it's a PAH that I used in some of my undergraduate thesis research, so I was familiar with it. And what we wanted to know is how was the PAH impacting monarch behavior. So in this study, there were 2 milkweed species. Plants were routinely fertilized with nitrogen on a monthly basis. Chronic exposure to CO2 for 7 months, ambient CO2 versus enriched CO2. And there were 2 AMF levels, plus minus AMF, and 2 toxin levels, the presence or absence of the polyaromatic hydrocarbon. And it's important to note that the toxin was introduced on 6-month-old plants, and then the monarchs was introduced when the plants were 7 months. So they were not chronically exposed to the toxin. So here's the experiment, and you can see that the monarchs, the monarchs are just doing their thing. So what happens is you have these monarchs in the cage, and they're hanging out all the way. They're hanging out in the back. That's where they stay. At this case, at this stage, they usually just fly out to get some nectar in their nectar dishes, and they go back. And then so at this point of the experiment, they have never interacted with milkweed. And then I would just introduce the racks, and then the monarchs could just slowly pick and choose the plants that they wanted to interact with. And the way this was done was by the way this was assessed was by doing sort of a temporal transect. So what I like to think of this as is sort of like, imagine if we all played musical chairs, around a few tables. And so we did that, and I played your favorite song for, 3 minutes. And then after I stopped the song, I scored and see who landed on a chair, what chair you chose. That's how we did the experiment. After 180 seconds, where the monarchs were at is where we scored. Sometimes they never left the plant. Sometimes they would hop to another plant. This was done for 4 experimental trials just to see if we could recapture some of the results. So as for the results, you're seeing that this was after 10 minutes of just using this sort of temporal transect to see what plants the monarchs were preferring. You can see when the phenanthrene or the PAH was absent, that they were actually more likely to visit those plants. And the ratio was about a 2-to-1 ratio, that they're more likely to visit plants without the PAH versus those with the PAH. And this pattern was also observed post 50 minutes. So post 50 minutes of the milkweed being introduced into the cage. Interestingly, this monarch association index was, as you expect, was strongly correlated with egg count, although we did not see that any of the main factors had a significant effect on egg count. We could also infer that over time that phenanthrene will be affecting the egg count due to the association, due to the linear correlation between monarch association and the egg count. Also, in this study, the plant growth promoting fungi did not impact monarch visitations, and neither did the CO2. And these are 2 of the experimental factors that were actually chronically or constant throughout the experiment. So for 7 months, the plants had associations with the AMF, and they had the CO2. And this is similar to our previous study where we showed that the biomass was actually depressed by chronic exposure to enriched CO2, and that AMF increased biomass. So we already know that the AMF increased biomass. This was published in Plants. We know that the CO2 decreases biomass, and this was going on throughout the experiment. And then so at 6.5 months, we introduced the phenanthrene, and then 3 weeks later, we introduced the monarchs. Although phenanthrene is known to slow plant growth, there's no reason to suggest that phenanthrene would have impacted the plant growth, because the plants had already established for 6 months. And in terms of the leaves, whether it's leaf area, the leaves did not fall off and then reestablish due to phenanthrene. Everything was already in play already. But although CO2 and AMF was not significant on monarch visitation or egg count, we observed a significant interaction. So what this means is that the circle and the square, they're not significantly different from each other. Neither is the circle and triangle is not significantly different from each other, and neither is the case here. What we're saying is that there's an interaction, and the simple way of observing this interaction is that under ambient CO2, the circle's higher. Under enriched CO2, the circle's lower, and the circle represents the presence or absence of mycorrhizal fungi. So it's just a significant interaction that we observed there. So PAHs are likely to cause stress in monarchs. Sorry for the typo. And the future direction that I'd like to do is I'd like to look at the microbiome of the gut. So here in the esophagus, I'm assuming that juvenile, which is not a butterfly, will be feeding on the actual milkweed, and it will impact the gut microbiome here, and perhaps we could see changes, because the microbiome is an important way of these different organisms to deal with toxins. But another way we could look at it is we can look at the role of oxidative stress. So these are the genes that were used in the previous study that I showed you with zinc. Superoxide dismutase might be an interesting one, as well as this one right here, GSTD1, which plays a role in antioxidant responses and detoxification. So that might be a way to look at this. So there might be different approaches, whether or not I put the phenanthrene in the soil again, or we actually put it in artificial feed. And so I'm talking with folks at Monarch Watch, as well as other scientists that are more molecularly inclined as to what's the best way to go about this approach. As for another future direction, as it relates to the early chicory study, for those who are affiliated with the KU Natural History Museum, it might be interesting to look at populations that are extinct versus those that are present, and seeing how they have changed over time in the local Kansas area. And also, one thing that I did not get to talk about is I did not talk about the this right here, which is the eastern hemlock, which is near endangered. But one thing I want to mention is that the eastern hemlock is near endangered because although I made it seem that near endangered species and invasive species, they operate in isolation, that's not the case. Sometimes invasive species could drive other species to become near endangered. And so that's the case on the East Coast with this invasive woolly adelgid, which is just wiping out hemlocks. So in Pennsylvania, you just see a lot of just dead hemlocks just standing there, and they're at the risk of just dying. And locally in Kansas and in Missouri, I believe that's to be the case with the emerald ash borer, which is doing some sort of the same effect. So I'm guessing in that scenario, it's an invasive versus a species being on the brink of and being near endangered. So as it relates to hemlock, I previously worked on hemlock, and what I can say based on implications is that although, you know, we like to think that the species being extinct or eliminated is just like, you know, bye-bye, sorry, we're bad, it's bad that that species is gone. But some of these species can also have biogeochemistry implications. So in this study, I was able to show that the hemlock soils actually had a slower rate of decomposition. So that suggests that eastern hemlock, their presence is also felt in the soil sequestration of carbon, carbon storage, and carbon cycling. So yes, there's an interaction between invasives and near endangered species. And I'm guessing there's an interaction where they could go back and forth. So in conclusion of this talk, what I showed you was that there has been genetic change in chicory since its introduction. I showed you that mycorrhizal fungi cannot provide bioprotection in the Colorado potato beetle and tomato pest plant dynamic. And then lastly, here's an area I'm starting to develop. I think that the study that I was able to show you was a study that I was able to get the results for over spring break. I think it actually leaves room for more discussion and more future direction. And one of the directions I would like to take is to actually look at the different antioxidant genes that are being expressed in the monarchs, as well as how it's impacting the microbiome of the midgut, or say the hindgut. And with that being said, I'd like to acknowledge some of the funding sources. So the Composite Genome Project for the chicory work, Monarch Watch for helping me with the monarchs and some of the future work that we've discussed. Also, Penn State Entomology for the Colorado potato beetles, the National Science Foundation, and the University of Kansas. And I forgot to mention NIH Aracta, which is my current funding source. And so thanks again.
So questions for Rondy?
Yes.
So where the AMF that did not prevent or reduce herbivory by the Colorado potato beetle, is that because the plants are more nutritious? I mean, what do you think the mechanism is, other than?
Yeah, so it's like a seesaw there, right? So there's the plants that are when they're with mycorrhizal fungi, or associated with mycorrhizal fungi, they're more nutritious, they have more phosphorus and more nitrogen content, which puts them at more risk to be exploited by the Colorado potato beetle. So that's usually a case where the plant association with mycorrhizal fungi is having a trade-off between better resource quality or associating with a fungi that can help prime plant defenses, because the mycorrhizal fungi, they infect the roots through the same or homologous process as, say, a pathogen, as some of the different roots pathogens that's forming Hosteria and infecting the roots. So mycorrhizal fungi, when it's forming the aspersorium and getting into the root cortex, it's similar to a pathogen. Only difference is by the time it gets to the root cortical cells and it forms its arbuscules, it's not robbing the plants of nutrients. It's actually fairly trading the nitrogen and phosphorus for the carbon, whereas a pathogen would just be depleting the host and not providing anything in return. And also the mycorrhizal fungi in that same process, they have the pathogen associated molecular patterns, which such as the chitin, which is universal in the cell wall of fungi. That in itself is able to activate plant defenses. So it's sort of like taking I don't know, a vaccine, if you will, right? So maybe the plant takes the mycorrhizal fungi, takes a vaccine, its defenses are on. So it can potentially provide bioprotection, but it gets very messy because the literature states that usually in that early form of colonization, there's defense genes that's being induced in the salicylic acid pathway. And then the defense genes in the salicylic acid pathway are usually good for deterring biotrophic pathogens, which are sort of like your gram-negative bacterial pathogens that don't really are trying to kill the host. They need the host to stay alive. So they're different from, say, a necrotroph, which is just trying to kill the host. And then the induction of those genes could deter phloem feeders such as aphids. So then that would make the plants more at risk to Colorado potato beetle because they're chewers and they're more likely going to be deterred by jasmonic acid genes. And now it really gets messy because AM fungi can induce those genes if it's a late stage infection. Now they're inducing jasmonic acid defense genes, and maybe that could deter the Colorado potato beetle. So it sounds like there's a lot of timing to do with the development of the mycorrhizal infection, which can impact these different defense genes. And then there's also the fact that mycorrhizae are making the plants more vulnerable to exploitation. And so it's very messy. Yeah.
Okay.
Yeah.
I believe you.
Other questions?
Yeah.
I'd follow up on that. Do we know enough about mycorrhizal fungi to know that there's different species that would be better perhaps for stimulating defense mechanisms in plants?
So I don't think we know that. I think all they've really shown is that the part about the different nutrient, the better ability to provision nutrients. So I don't think there's any really much thing on just the better defenses, because what the plant is recognizing, whether it's the patterns that are similar to a pathogen, is conserved universally. It's just conserved. So I don't think there's really an advantage to the different types of I don't think there's an advantage, and that probably explains why we saw that all the other fungi, they were all behaving in the same direction. And I guess if they weren't, maybe we could say it's a nutrient effect too, because I guess the nutrients, there are cases of some mycorrhizae being better at provisioning nutrients. So yeah, so and I guess that gets back to your question that it could be sometimes a nutrient effect. Yeah.
I had one, while thinking about your multi study, you had that nice picture of the pipeline in the background. I'm wondering if you know about, has there been any work in the wild on like looking at the plants that established, like for example, up in oil spill, when it's happening in Kansas or
Mhm.
on Dallas or something. Are the plants that grow in these spaces then terrible hosts for, monarchs, for example, or of any sort of work in that realm?
I can't think of the PAH work, but I have heard of previous work at KBS of folks looking at very saline soils due to extraction of, I think, what was it, gas, or you probably know it better than me, the extract, just trying to remove some of the oil and gas. And then so
Yeah.
by doing that, you're able to put a lot of the salt from lower horizons are now reshuffled, so now they're on the higher horizons, and then now you get salt stress from trying to extract the oil or gas, if you will. And then they've been studies by faculty at KBS, like in the 2000s, where they tried to look at different plant tolerances to the salt, and I'm guessing as a mechanism of phytoremediation. So then you could remove some of the things that are causing stress, such as the salts, and that would allow the monarchs or a different population to do better. And I guess that might relate to, while putting the salts from the lower horizons on the top horizons, that could probably relate to zinc, which is one of the papers that I had showed earlier on, how just zinc toxicity can lead to stress in the monarch. So so I'm guessing, if I tie all the points together, that I think that it will impact, those will impact monarchs just through trying to get the oil and gas, even if it's not directly, the PAH. There's so many other anthropogenic stresses that just goes into mining for oil and gas that I can imagine that there will be ramifications.
Well, if there are no other questions, let's thank Rondy again.
Thank you.